Hello. Hello, am I audible?
Hello, Miss Dr. Banerjee. Hello.
Hello. Yeah,
Yeah.
I cannot see anybody else's screen, so
Oh,
am I audible?
oh, really? Maybe you can setting the.
No, I mean I can see the screen, but nobody is online, or maybe the video is off. So I'm not
Oh,
if the
okay,
meeting
okay.
has already started or not.
Yeah, we already started. Hi, Swati. This is
Hello.
Macro. Hi.
Hi, Mikro.
Hi,
Hi.
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 Ho-
How are you? How's it
Yeah,
going?
yeah, everything fine. And how are you?
Okay, me too. I'm fine, thank you. Yeah, you already seen the screen, yeah, Swati. We
Yeah.
already
Yes. I
yeah
can
have
you hear me
45
and see me?
participants now. Yeah. We
Okay.
hear you clearly,
Right.
and I think it's everything is okay.
Okay. Right.
Okay. Yeah.
Okay.
You
So
ready?
good. Yeah,
You
of
ready?
course.
Yeah, of course. Always, as always. Yeah.
Yeah.
Okay.
So
Thank
okay.
you.
So good. Yeah. Right. So I will start with saying good morning or good afternoon
Yeah,
to. I
before
think.
begin, I think I should to introduce. We will guided by Rocky Alfanz.
Okay.
He is my colleague
Hello.
from
Hello.
electrical engineering.
Right.
Yeah, I think the time is yours, Rocky. So he will be collected all the question and guide all of the event today, Swati. Thank you.
Okay.
Okay, thank you, Mr. Dr. Mikro, for this session. Okay, good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Waalaikumsalam
Uh,
warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi.
okay. I'm Rocky Alfan speaking, and I'm representing the International Office as moderator on the valuable research talk now. And well, before we begin the occasion, I would like to send my deepest grateful to the team of the International University Sultan Ageng Tirtayasa for having this event. Okay, this presentation is about to begin. May I have attention, please? And I proudly introduce Honorable Dr. Swati Banerjee as our main speaker, who will discuss human-centric computing toward healthy living. Well, before the presentation begin, allow me to read briefly Dr. Banerjee biography. She's a PhD degree in biomedical engineering. Now, Dr. Banerjee activity is researcher of Axi Marseille University. Dr. Banerjee have interest with the research of biomedical signal processing, machine learning, computer vision, and pattern recognition. Okay, we will hear the presentation maybe approximately 20 minutes or more. We will continue Q&A session, maybe about 10 minutes. And if we still have time left, we will extend the Q&A session. In addition, you can also send us a question using the chat box on the Zoom. All right. Please welcome Dr. Swati Banerjee. The floor is yours.
Okay. Thank you so much, Rocky. So I will take this opportunity to thank Micro and also being patient with me because I think I rescheduled it once or twice, and apologies. So Micro is a very dear friend of mine, and like I wanted to give the presentation like not. I wanted to focus more on a broader area, broader aspect of like how biomedical engineering can shape our health and our life, and not to bore you with my research work, which is like very specific. So okay, so I will share my screen, and please let me know if you can see it. Is it visible?
Yes, it's clear.
Yes,
You know.
yes.
Yeah.
And do you see it in a presentation mode, or
Yes, I
Yeah.
see the presentation.
is it in the presentation mode now? Is it good?
Yeah. I think it's
Yeah,
good.
Yes,
I think
good,
it's good.
good, 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 good.
Okay, right. So, as already being introduced, I am working as a research engineer in INS at Axi Marseille, and my primary area of focus now is neuroscience. But today I will be talking about human-centric computing toward healthy living. So I decided to give a holistic overview of the scenario and the various type of biosignals that can be used to make human life better. And so I am going to walk you through some of the CAD-based systems, and for which I have broadly I will be like moving around these four topics, which is like how we can detect cardiac abnormalities using using signals like ECG, heart rate variability, heart sound, etc. uh, how can we estimate, or how can we predict, or how can we give a prior uh, like indication to a subject that the person is going through psychological stress by analyzing the pulse rate variability or PPG morphology features. And then I will like to discuss a little bit about the neural mechanism through auditory pathway, or how it can be done, and the evaluation of the motor function age that can also be done by high density surface EMG signals and acceleration data. But before these are like two topics which are too like focused. But before going to that, I would like to talk about the nature of the biomedical signals. So as we know, like living organisms are made up of many component systems, and each system are made up of several subsystems. So each have a very definite or very specific physiological process going on in the background, and they actually are the manifestation of the nature, or the phenomena that is going on behind, and those phenomena can be modeled by like physical systems or dynamical systems. But anyways, I'm not going to talk about dynamical systems in this talk. What we can do is we can identify some kind of disease or defect in the biological system. They are more like to understand if what is the variability in a normal or a healthy cohort, because sometimes each human being is one entity and they are different. So even in healthy condition, people can have different clinical signatures. So we will also like there is a huge amount of work going on understanding the normal or the um, non-pathological behavior of the subject. And pathology is also involved, which is kind of a very different game altogether. So this is kind of a biomedical signal analysis framework, where if this is the patient, can you see my mouse?
Sorry,
Can
Ms.
you see?
Banerjee, your slide
Yes.
is freezing, so we can look your slide clearly.
Is it freezing?
Yeah, I mean your slide is not next, something like
Yeah.
that.
You
It's
are
not.
still in the first slide,
Yeah, you're
Swati.
still in the
Ah,
first slide.
Yeah.
okay. So sorry. Um, is it good now, or no change?
Not yet.
Okay, just a moment, please. Can you see now?
Not yet. We are still
Okay.
waiting.
Um, now.
Yeah, it's great now. Yeah.
Okay, so what I'm going to when I put it on the presentation mode, there is a problem. So I will go ahead with the slide mode, maybe,
Okay.
if that is
This is
okay.
great.
No problem.
Yeah.
Okay, so I was already on the third and the fourth slide, so maybe we should start from here again. There was not much that was to be shown in the previous one, just a recap. Okay, so yeah, so this is like a kind of a framework that I have put into, and most of okay, it's visible, right?
Yeah,
The
now
slide
it's
is
great.
visible.
Yeah, now it's
Okay.
great.
So most of the time people like we have kind of some sensors which send signals, and those sensors can be like bedside ambulatory sensors, or they can be more like handheld devices. For example, we use Fitbit or some other devices, or like nowadays there are people who are suffering from brain diseases where we use deep brain stimulation, where the devices are implanted into like deep structures of the body. So this is also one application. So when people say that they are working on biomedical signal analysis or biomedical engineering field, is quite a huge and a vast area, and there are multiple things that we can, as a researcher coming from computer science or electrical engineering or electrical engineering background, we can contribute. For example, building the infrastructure is one thing, and then also we require some like we need to analyze the signals in real time sometime, so we require faster algorithms, and this is kind of a framework. So I will go to the next slide. So what are the objectives? They are more like information gathering, diagnosis, monitoring, therapy, control, and evaluation. And as I already said, signals can be acquired in an invasive or non-invasive way. It can be yes, please.
Sorry, Miss Banerjee.
Yes.
Maybe you need to share your screen, mirror screen. I mean, because not full screen looks like.
I don't understand. Should I stop sharing and reshare it again?
Uh, yeah, I think it's great. Yeah, the point is we see your
Yeah,
presentation clearly. It's great. It's good, Swati. Yeah, if you can make mirror mode,
yeah,
maybe it
mirror
will.
mode.
Yeah, so we see clearly. But I think it's okay.
Yeah.
We don't want to interrupt your
Okay, okay, sorry.
mind of seeking.
Yeah,
Yeah, yeah.
No, it's fine, but you have to
Okay,
guide me a little bit how to do the mirror thing, because I
okay.
have
No, I think you
so
can continue with previous method you already know, so it will be easier for us.
okay.
Okay. Yeah.
okay, let's continue. It's okay.
I'm so sorry. So you can see my screen, is it or not?
Yep. It's clear.
Okay, right. So this is like I was talking about the acquisition procedure. There are multiple acquisition procedure, and as we acquire the signal, noise can, we know, like the more we go away from the source of the signal, the more the noisier it can be, and more the clinical signatures are becoming hazy. So we need to have multiple algorithms in each of the level. For example, if we are doing kind of deep brain stimulation, we will have our electrodes in the subcortical region of the brain, which will give us more like signals which are more prominent, more near to the action potential, or whatever local field potential kind of signals. But when we go to the, for example, Emg, we are basically measuring the like from a global point of view, global perspective, and those clinical signatures will be very, very different. So we have to model, or we have to model the signal in that way, or we have to devise new schemes to find a concordance between various level of acquisition. So this is why I wanted to put this slide to make this point. And this is a common like common framework which is being used, and which we have a patient side. We can work for building more and efficient transducers, so that the noise cancellation is better, the signal acquisition is better, etc. Then we have amplifiers, etc. And then we convert the signal, we filter them, detect events that are clinically relevant. Some, not all events clinically relevant to the clinicians are can measure something. So as signal processing experts or engineers, we need to have efficient features extraction algorithm, which will give us not clinically relevant, but they are pseudo features which will guide our algorithm to make a more knowledgeable, more like informed decision when we are going to diagnose something. So here comes our role, and then we all know why CAD systems are used. And what are the hurdles? The most of the hurdles come for the automatic analysis of signals. They come from noise, because the noise, we don't know what is the nature of the noise, from where it is coming. Sometimes the noises are actually internal bodily signals. They pose as noise. For example, if I have a ECG signal, it can even it can get like contaminated by EMG signals, which are actually noise in context of ECG signals, but they have clinically relevant information if we look at them from other perspective. So there is always a trade-off. What is noise and what is not? How should we classify noise? Or even if it is a noise, or if it is actually a manifestation of some other biophysical phenomenon that is going on. I can cite another example, which is which we get in ECOG, where there are some action potential like spikes that we can see, for example, before seizure onset or something. So those action potential or those jumps are basically coming from the cortical surface of the brain, which are actually manifestation of some underlying physiological phenomena or physiological activity that is happening. But there is a huge debate between researchers. Should we consider them as noise or just completely discard them, or they are actually manifesting something else, which will further detect or further like is kind of an attractor which will further launch some other phenomena? Or is it a cause, is it an effect? We don't know. So we often use like strong machine learning algorithms to quantify the feature or the algorithms that we have developed. And then associated complexity is also an issue, because if we need to prepare our algorithm to be adaptive to a handheld device, it has to be faster, low in complexity, low in terms of space and time. But then if we have time and we can use supercomputers, we can give our computation to like supercomputers, and they will do the job. So there is also a trade-off in this manner, and there is huge research going on what to do, like in simulation science, how to use it, etc., etc. So I think the video will not play. I'm so sorry. So I will start with very basic how the action potentials look like. They are like one on cellular level, and you can see like this is the origin of the biomedical signal. Well, like the cells are like dipole. I have taken an example of a rabbit arterial myocyte, which this is how the action potential will look like in time, and it evolves. And when we have a collection of cells and a collection of like it can be like neural population, it can be myocytes, whatever, and they will manifest themselves as random signals, which are very like they are not repetitive. They can be repetitive, they cannot be repetitive. So these are some of the biomedical signals like ECG, EMG, EOG, and we know how they are acquired. So there are some of the noises that I have already talked about, and two other type of noises that I did not talk about are like electrode contact noise and electrosurgical noise. These type of noises are they are very hard to detect, because it's easier to detect if the acquisition system is non-invasive. But in case of invasive system, we don't know if the noise is electrosurgical noise, or usually the electrode contact noise is very difficult to detect in case of deep brain stimulation. For example, if there is an implantation which has been done inside the brain, we really do not know what is creating the noise. As I have already said, that if it is a noise, should we discard it, or it is like a physiological phenomena. Okay, so this is our cardiovascular system. Maybe we have already seen it before, and this is, if you look at it, it's kind of a like the circulatory system can be modeled as an electrical circuit, or people do in control system, they also model them, but I have kept the modeling part apart in this, so we will not talk about it. But these are the cells, and these are responsible for generating the electrical pulses that we have. So this is just a conduction system of the heart, where just to make you familiarize about how the heart actually works. It's like a pump. If we can think in terms of engineering terms, we will say that it's a pump which is getting initiated by the sinoatrial node, and then all the process, all the laminar flows, whatever type of flow is inside, it's being initiated by it. So this is how researchers do. They just try to limit, because in case of biomedical signals, as I have said, things are not predictable, but we try to take away some of the unpredictability, and from what we know from our engineering knowledge, we try to model the system in that way, and we try to understand how the working phenomena, or if we need fixing. For example, sometimes there is myocardial infarction, then the flow will change, and we will get a corresponding different clinical signature in the electrical recordings. So this is the physiology of the contraction, where we have a depolarization, plateau, and repolarization of the cells, and this will actually give us the ECG signal that we will see later on. And I am not going into the detail of action potential, how it works, but if somebody is interested, maybe we can talk about it later. And this is how the heart can generate one ECG cycle. So this is a collective signal where we can see how the action potentials are from each of the region. They are very different in morphology, they are very different in shape. And if we think of the signal as a pattern, then we can see all this collective pattern is manifesting itself as a very beautiful, very rhythmic ECG, one cardiac cycle, which is represented in a one ECG bit, which is like the P, QRS, T, and U. At this point, if someone has any question, so I can. Okay, so we will go ahead. So this is one ECG cycle, and then this is how it actually looks like. And each of the chambers of the heart, we can see like it depolarizes and repolarizes. So basically, any change in any of the morphological feature can actually represent some kind of underlying pathology, or some kind of disease that can be identified by analyzing the signals. So this is how a 12-lead ECG placement system looks like. And we can see like now we are coming into a three-dimensional plane, three-dimensional system. So what we can do, we can actually diagnose something. We can do some kind of functional analysis to understand the rhythm of the heart better, because as I said, there will be a variability among healthy subjects as well, and there can be some implants that can be made to make our like some ailing heart better. There can be like we will have biofeedback, and they sometimes are used as biometric. So there are multiple applications. And we can also, as like in kind of sports medicine, etc., people do peak performance training and monitoring. So these are some of the applications. So now I will talk a little bit about EMG signal. This is kind of a surface EMG signal where we can see that there is a contraction and there is a relaxation phase. So, so this can also, like if we analyze EMG signal, if you see that it's not a very rhythmic signal, but we can also get some kind of information if we try to extract the envelope of the signal. So the thing is, mostly EMG signal, it's an invasive procedure. Till now, it's in the trial phase. When we analyze EMG signal, there is a huge debate whether we should go invasive or we should remain non-invasive, because as you can see, we cannot actually find out any pattern if we just try to do a point-to-point correlation. So we have to use higher order statistics or higher order signal processing techniques to extract and squeeze the signal to get something out of it. So there is a research that is going on to understand the sarcopenia, which is like understanding the aging phenomena better. So sarcopenia is nothing but muscle loss. We really do not get to understand when a subject is going to lose the muscle at what age. So by analyzing this kind of signals, is there a possibility that we can actually say the person is perhaps not very active, or the person has like need to flex the muscle more, or something, to do some sports or something, so that the muscle loss in old age doesn't become a problem for them. So this is another area of work that is being actively pursued at the moment. And there are multiple devices. This one is practically non-invasive. And these are some of the uses of EMG, like EMG application. They are rehabilitation, functional analysis, as I already said. And for prosthetic people who like war veterans, when they come back from sometime they have to be amputated. So they need, if they are going to be attached with some prosthetic limbs or anything. So there also EMG finds a very like prominent role, where it can say how the not only the muscular activity, it can also connect our body or our muscular movement to the autonomic nervous system, and it can say a lot of things. So here comes the king of the signal, I would say, the EEG signals, which is coming from the brain, and that's kind of a muscle, but it's like a very different muscle. We often say that like the brain muscle is working, but actually, if we see the phenomena is very, very different from the EMG. Although if you see like the signals are equally random, they are generated like equally random, they are not rhythmic at all, but still. And this is like a non-invasive EEG electrode cap, and there are multiple ways by which we can divide the scalp and equate the signal. This is a particular 10-20 system that I am showing. And these are the rhythms that are the dominant frequencies that we can see. And each of the delta, theta, alpha, and beta band of the signal can say different brain rhythm, like about the brain rhythm and what is going on. Like what is going on behind the scene, and how the brain is working. Is it like in a deep sleep stage, or is it like in a sleep stage? This type of things can be very helpful to analyze when particularly subject having kind of disease like Alzheimer, or sometime when we do deep brain stimulation when people like with people having epilepsy. This type of phenomena, I mean, these theta propagation, the alpha theta propagation, how the brain rhythm changes is also an area of interest. And of course, we have the EOG signals, which are, as I said, what we are looking, how we are looking at the signal and what is our goal determines if the signal is a noise, should we consider it as a noise, or is it something which is of importance to us. For example, in an EEG, we will consider this eye blink movements as noise. But for example, if we are trying to do some kind of visual attention task where we need to see what is the fixation, what is the how much is the, what is the eye movement, how much the person is actually looking at something to make a decision, these eye blinks or these gaze things are becoming very important. Again, the forehead muscle tension, which is which also is an important phenomena for facial recognition or kind of things, to if we want to understand or if we want to automatically detect if a subject is getting very stressed. For example, if a driver who has been driving for like 2 days, we can understand from the muscle tension if the person, so it's kind of a, it's kind of a neural mechanism by which we can train, or we can also do that with monkeys. The way the monkeys are being trained, they are given visual cues, and when they do something, they are rewarded with some kind of food. So it's kind of associative memory that is being getting worked upon, and this can happen. So
It's true, huh?
yes, it's true.
Yeah. Okay. Thank you.
Okay, let's continue. There's any question, this audience?
I see many people like complaining about the screen. Please accept my apologies. It wasn't, I don't know, somehow in my computer when
Yes,
so
I want
Okay.
us.
Okay, please. Your name, please state your name and do the question.
My name is Farizal. I want us
Okay.
to Miss Swati. In addition to the tools described earlier, are there simple tools that can be used in biomedical, such homemade experiment tools or other? Because I know biomedical tools are a bit expensive.
Thank you for a question. I would like to come back again. So you by biomedical tools, you mean like the acquisition system, or what type of tool are we exactly talking about?
Maybe like biomedical equipment is very expensive, maybe for the price. So
Mm,
yeah,
yes, I mean,
yeah.
it depends on what type of research you want to conduct. For example, the last slide that I showed, which is like, I kind of like the signal very much. It's a PPG acquisition device, which is not very expensive. And nowadays, people can do it just like using the camera of your mobile phone, and you can have the signal because it's just a diode which is like measuring the change in your blood volume. So it depends on the application. And of course, if you want to do, for example, deep brain stimulation, that is very expensive. Not only the device but the procedure, because you will need a team of surgeons to open up the brain and then put things inside someone's brain. It depends on the application. Yes, I don't
Yes,
know if I answered your question or.
Mr. Fahrizal, you understand? Hello,
Yeah.
you
Did
get
I?
your answer? Hello?
Yes, I think
Yeah.
enough.
Okay. Okay, there's anyone want to ask question? Hello? Okay, maybe I'm
We
as a
have in the
moderator.
chat question. I think.
Yeah, chat question maybe. Okay. This from Mr. Imamul Mutakin. A quick question. Instead of the measurement signal from brain, it's possible to actually stimulate the brain with some kind of signal to manipulate to the act or behavior.
Okay, this is a very interesting question and a very tricky one as well. So when you say measuring from the brain, do we mean like measuring it from how do we want to measure? It depends on if we are going to measure it like invasively or non-invasively. So if it is a non-invasive measurement, it's more like EG signal or MEG signal that we are measuring. So in those type of, when we do non-invasive measurement, the stimuli should come from eye or other sensory organs, for example touch, or it can be some auditory cues like we are listening to some good music. and and when you say thought, I will if it is non-invasive, most of the time people try to see what is the behavioral change. For example, if someone is being uh, like you stay in a very calm and quiet environment where you are listening to a very soothing music, and after that it has been proven that if the person is like they get focused enough to be able to solve complex mathematical tasks. So this is one area of work. This is the non-invasive thing that we can talk about. But if we talk about invasive stimulation, so in that case it will not be some visual cues or auditory cues. We will not be listening or we will not be hearing something, but they will be actual kind of electrical pulses that will be put into the brain regions to make them activate, to make the neuron population activated. So in that case it can change the behavior in a way. For example, some person, they are having Alzheimer's, or they are having, maybe they had an epileptic fit, and after the epileptic fit they are having kind of convulsion or something. And if we stimulate the deep brain regions like the subcortical regions, which are like the hippocampus, the amygdala, etc. So hippocampus is known to have a it's kind of the memory hub of our brain. So if we stimulate it, it can sometime sorry for the noise, it can sometime make changes in the neural circuitry. So that is another type of change, and we don't know what type of behavioral change we will be able to see in the subject. It can be for good, like they can stop having the epileptic fit, or they can like because of the perturbation of the brain network for that particular moment, uh, maybe they are having some kind of memory loss which can be regained back by stimulating the hippocampus. So these, I can just like to answer your question, I can think of two ways. One is non-invasive, one is invasive, and the stimuli depends on if we are doing it invasively or non-invasively. So we have to pass the stimuli through something. Um, is it? I think did I answer the question? So
So it's okay. Okay. Yes. Thanks.
okay.
Okay. Is there anyone want to ask question?
May I?
Okay.
Yes, please.
Let's please.
Okay.
Is there
Thank
your
you.
name? Okay.
My name is Yayu.
Okay.
Thank you for your presentation, Swati.
Yes, my pleasure. Thank you.
Okay. I'm very interested with your explanation on the noise. How to differentiate the noise with the true signal.
Right.
I'm working on when you're working with human, I'm working with plants. So I measure whole plant photosynthetic rate invasively. in a commercial greenhouse, and
Okay.
um, I would like to know how the process of the signal to come to maybe in your area is a decision whether you can make a medical diagnose based on those signals. Can you explain more? Maybe uh,
Um,
how is the process?
you okay, so what I understand, you are measuring plant signals from the plants,
Yes,
or
photosynthetic rate. Maybe it's like ECG in your area. Maybe. Yeah.
okay, so you wanted to know about how to make a prediction
Yeah,
about something.
I
Is
think
that
Yes, a model plan diagnosis,
yeah,
something like that.
yeah.
okay? So
Especially Swati, maybe what I think of is like how you make filtering about differentiate the good signal and the noise
okay.
and can absorb it. That's why.
Yes, thank you.
Okay, right. So for that, because I have not, I'm not an expert. I mean, you're an expert in that field. So can I ask you, what type of noises do you think is inferring while you are measuring the whatever you are measuring? So what are the noises? Do we know about them? Do we know the characteristic of the noise that is like interfering? Is it kind of an external noise, or it is an internal noise of the system? Do we already know about those things, or is it a very new area of research? I don't know. I mean, you have to help me in this.
This
Mm,
is quite new in the
okay,
area.
right.
So we are measuring the photosynthetic rate with
Mm-hmm.
of a whole plant of cherry tomato inside
Mm-hmm.
a chamber, and
Okay.
then we got the rate of the photosynthesis activity of the plant,
Mm-hmm.
but we are not sure because there are sorry for the noise
No
because
worries.
this is quite new. We don't know which how do I say the current photosynthesis is normal or not.
Mm-hmm. Oh,
Yeah. So
okay.
how do you make
and and did you
analyze? Yes.
I'm actually very impressed about this thing, because I have never heard about it. It's really something new to me. So I'm very interested to know, is it a homegrown sensor that you are using, or is it like something commercially available that you are using, or did you made the sensors? How the sensors look like?
Okay, actually, that's my PhD study, so it's
Okay.
teamwork. And what I do is I process the data and
Mm-hmm.
make analysis for it. Now it
Mm-hmm.
comes to the how do I say the techniques in the processing, but we
Mm-hmm.
haven't come to a decision whether this signal is normal because
Mm-hmm.
we do not have any data showing
Mm-hmm.
the
How
normal
it
photosynthesis. Yes. So
exactly, this is
yes,
this is
yes.
often a like huge problem in case of new technologies, when we try
Yes.
to roll out new technologies. So what I can think of at the moment is, maybe you can do spectral analysis, and then if you have multiple instances, when you do like the spectral analysis, you can try to do some spectral pattern matching. For example, over a period of time, you can see which patterns are consistent. For
I see.
example, if you have a signal for maybe 20 days, you cut them into epochs of 1 day, and each for 20 instances, you do the spectral analysis. We do you do the You can take like s- simple wavelet based analysis or whatever, and then you can try to see, when you're getting those spectral features, you see like, over the 20 days period, which are there some features which are consistent. So and then maybe you can also do a Fourier analysis to see what are the spectral components that you have in the signal. And then when you do the Fourier analysis, and when you do the wavelet analysis, and then you can make a comparative study, that which are consistent, that you separate them. Those are one type of features. And then there are some features, maybe they're evolving in time, maybe they also have some kind of like they will in future they are going to manifest themselves into something, but we don't
Yes.
know at the
Okay.
moment. So you can try tracking changes in those type of signal features, and see how they are evolving over time. So this way you can actually separate both of them, and then consistent feature and inconsistent features, and then from the inconsistent features you can try to gather some more information, squeeze things, and then see how it is evolving. I think
Okay.
it will
Thank you so
be
much. Yeah, that's helped a lot. Yeah. Thank you.
okay.
Thank you. There's any question in this audience?
Thank you. Maybe I
Thank you.
want to add some. Swati, thank you for your great insight. And I just
The pleasure
think,
Yeah.
is all
yeah,
mine, Macro. Thank you for inviting me.
yeah. I think we can conduct maybe in the future. You have a lot of practical knowledge to how to analyze and to filter it. I think it's quite interesting. And honestly, I don't see we have prominent researcher in this area in my country, in Indonesia, I think. And especially what Ibu Yayu told to us that it says we don't have any special lecture to do that. We really hope if you interested in the future, we can buy like some short course or something like that.
Research
Uh,
collaboration, I think.
yeah,
Yeah.
Of
collaboration,
course.
something like that to equip our university, especially in Untirta, where I am working now. And we really looking forward for your great contribution as always. Thank you,
Yeah.
Swati.
Thank you, Ms. Sarwat. Okay.
Okay. I yes.
Finally, we come to the last part, yeah, of this research talk event. So we would like to say thank you for Ms. Dr. Saraswati to present the informative, interesting, yeah, talk to audience for the active participation. Hopefully, this presentation will be beneficial for everybody. Thank you for attention, and maybe this after uh, we got uh, we will see another maybe collaboration with your institution, maybe, or we
Yes.
have
Right.
some
Of
lecture
course.
for interesting research. Yeah.
Sure. I
So,
would be very happy to
yeah.
be able to collaborate.
Yeah,
Yeah.
yeah.
Or
Collaborate.
if
Maybe visit us
someone
here. You're very
yes, that
welcome
would be
in
amazing.
Yeah.
Indonesia, Swati.
Welcome.
I did not ask how is the COVID situation there? Is it like okay, under control? Everybody is healthy and like
Yeah,
this?
Yeah.
yeah. Go
Okay.
ahead.
Okay.
Okay.
Thank you, Miss
Yeah,
Saraswati
yeah.
Banerjee. Thank you for your
Okay.
session.
Right.
Okay, yeah. Just for the closer about the COVID, I think in the all of the world, I think we are facing the same thing for
Yeah.
Hmm.
today. But we hope and we believe that we will pass through it in the future. So we really want to keep in contact with you, Swati.
Of
And
course.
wherever
Mm-hmm.
you go, either
Mm-hmm.
stay in French or go back to India. So uh, yeah, we really hope we can have mutual collaboration with you. So stay
Right.
safe and keep
Stay
in contact.
safe.
Yeah.
Yes, of course.
Okay. If you have any closer statement, please, Swati.
Well, no,
Yeah.
it was an absolute pleasure to be able to talk and see Macro after so long. So it was really nice and a experience for me as well. Thank you for inviting me, and yeah. Stay safe, stay healthy.
Stay healthy too.
Yeah.
Okay. See you.
See
Thank
you.
you
Okay. See
all.
you.
See
See
Bye
you.
you.
bye.
Bye.